Okay, hello everyone. Thank you for joining us today in Thursday webinar series. My name is Esra. I'm at the beginning of my career and very happy to have a chance to moderate this wonderful seminar thanks to Open Back Science. And we have two excellent speakers today. We are going to talk about SARS-CoV-2 scientific updates. And I want to introduce our first speaker, who is Felix Pahmeier from Paderborn, Germany. He received his Bachelor of Science in Life Sciences from University of Münster and currently conducts his master thesis research in the lab of Ralf Bertenschlager at the Molecular Virology at Heidelberg University. He was in the US Embassy last year on the day the former president announced a travel ban for Europeans due to COVID-19. When he came back to the lab, he started the cloning for the SARS-CoV-2 reporter cell line. He is now going to talk about his project, a versatile reporter system to monitor virus-infected cells and its application to dengue virus and SARS-CoV-2. So please share your comments and questions. We are very excited to hear more about this research. So let's hear. Felix.
Thank you, Esra, for the nice introduction. So I hope you can hear me well and see my slides.
Yes.
Okay. And once again, thank you. And also thank you for the organizers for inviting me to talk about our work for the generation of reporter cell lines for the identification of dengue virus and SARS-CoV-2 infected cells. So as a bit of background, I will be focusing on RNA viruses and specifically RNA viruses that possess a positive sense single-stranded RNA genome. To this virus group belong, for example, the Flaviviridae, to which dengue virus and Zika virus belong, or also the Coronaviridae, and the most well-known member now is SARS-CoV-2. So briefly, for the life cycle of RNA viruses, after entry, mostly by receptor-mediated endocytosis, the viral RNA is released into the cytosol after fusion of the viral membrane with the endosomal membrane. Since the RNA genome has a positive sense orientation, it can be recognized and translated into polyproteins. And these polyproteins are then processed by viral and host proteases to generate individual proteins. Furthermore, the viral RNA is then amplified in complexes in the endomembrane system, visualized here, for example. And this network of rearranged membranes allows the efficient replication of the viral RNA. Finally, the new virions are assembled containing the amplified RNA and then released from the cell. So I talked about now the intracellular life cycle, but how do we even know what happens inside the cell? And more importantly, how do we identify cells that are virus infected? So it's sort of similar to what happens with people that are infected with SARS-CoV-2 or with viruses. We mostly know only when they show symptoms, or in case of cells, when they look bad and are dying. So to circumvent this problem and identify cells already at an early stage, different approaches can be used. One of these approaches is the generation of a reverse genetic system. So basically, the genome of the viruses is synthesized on a plasmid, and this plasmid can be introduced into cells, thereby generating a recombinant virus. And these reverse genetic systems can replace certain areas of the genome with fluorescent markers, for example, and thereby visualize which cells contain the RNA or the genome and are therefore infected or transfected. While this is focusing on the virus side, also cell-based assays can be used to identify cells that are infected. And one I've shown, and I'm showing in an example down here. It can, for example, be visualized by using fluorescent signals that change the localization, for example, here from the ER to the nucleus. But how do we specifically only find this translocation or target this translocation only in cells that are virus infected? Well, I previously mentioned that during the viral life cycle, when the RNA is translated, in case of both flaviviruses and also coronaviruses, this involves the generation of a polyprotein. This polyprotein, for example, visualized here for the flaviviruses, can then be cleaved by, in case of flaviviruses, the viral protease NS3 together with its cofactor NS2B, and also in case of flaviviruses by host proteases, thereby generating individual proteins. Additionally, for coronaviruses, they express two proteases located in the non-structural protein 3, down here called PL-PRO, or in the non-structural protein 5, which is termed either M-PRO for main protease or 3CL-PRO. So importantly, these viral proteases recognize specific sequences in the proteins where they target the cleavage of and thereby release the individual proteins from each other. So we thought we could utilize this activity by designing a reporter system that is based on 3 modules. First of all, we have a fluorescent protein that is fused to a nuclear localization signal. And the next module is a protease cleavage site, so a linker containing the protease cleavage site. So this is basically the sequence that we find in the viral polyproteins, for example, that is recognized by the viral protease. Finally, we have utilized the ER resident protein Sec61 beta and specifically only the transmembrane domain to target this construct to the ER where the polyprotein production takes place. So this is a scheme of the predicted topology of our reporter construct, and you can see the ER anchor here, and then in gray the protease cleavage site. So when the virus is present, or specifically the viral protease is present, this cleavage site would be recognized and cleaved, thereby releasing the fluorescent protein from the ER. The NLS that is fused to the fluorescent protein would then translocate to the nucleus, thereby allowing to identify specifically which cells are either virus infected or have expression of the protease. Additionally, we designed the construct in a way that restriction enzyme recognition sites are located on either of on the sides of each module, thereby allowing, for example, the exchange of the fluorescent protein for different colors, and also the adaptation to other viruses that might have different protease cleavage site sequences. So in the first step, we were interested in generating a reporter cell line for dengue virus. And as mentioned previously, in case of dengue virus, the viral protease NS2B3 is responsible for cleaving some of the non-structural proteins. And we inserted these sites that are distributed through the polyprotein into our construct, transduced cells with the construct, and then infected them. And what we show here is exemplary images of these cell lines and then a quantification. And in case of the uninfected cells, what we observed was that some constructs showed already nuclear translocations as quantified here with the green signal already in absence of the virus, indicating that this might be either mediated by a cellular protease or that this is unspecific. This was not the case though for protease cleavage site construct number 1, containing the cleavage site of between capsid and PRM. And what we observed was that upon infection, so we stained here a viral protein in magenta, and when we quantify this, we saw a high number of cells that were positive for the viral protein, also positive for nuclear GFP signal, indicating that our reporter construct can reliably detect virus-infected cells. So we wanted to further characterize this reporter cell line and did a time course experiment, ranging from 24 to 72 hours. And then we stained the replication intermediate double-stranded RNA. And what we observed was that we have a good correlation between the staining and our reporter cell line, and also an early detection as early as 24 hours in this experiment. Furthermore, we wanted to check if the other serotypes of dengue viruses, so 1 to 4, are all, so the previous experiments were done with dengue virus serotype 2, but also our cell line was able to detect infection by dengue virus 1 to 4 with our number 1 construct. We then combined the reporter cell line with the previously mentioned other approach. And we combined it with a recombinant virus that expresses a fluorescent protein. And you can see the signal for the reporter virus here in magenta. And what we observed when we quantified this is that we see a good overlap between cells with nuclear GFP signal from our reporter cell line and also nuclear fluorescent signal from the reporter virus, indicating that this is really a specific translocation only occurring when the virus is present. What also was interesting is that the wild type virus showed a higher and earlier percent of totally infected cells, indicating that what has been shown already for other viruses, that introduction of a fluorescent protein in a virus genome might lead to a reduced replication of this recombinant virus, which is an advantage of the reporter cell line, where you do not need to modify the virus itself. We then were interested in how early we can detect infection, or in this case, we used a expression-based plasmid, and this has been described or published last year as well. It is basically expresses the polyprotein of dengue virus and can induce the replication sites in the absence of viral replication. And when we did live cell imaging with this construct, as early as 6 hours post-transfection, we could already detect cells with nuclear GFP signal. So I will play this once again. Now you can see that these cells are positive, while the mock cells show that the signal is retained at the ER, indicating specificity. Then the pandemic happened, and we were desperately looking for tools to use to study this virus, and especially look at the intracellular life cycle. So we basically were interested if we could adapt easily our cell line or our reporter system to SARS-CoV-2, and therefore introduced the sites, the cleavage sites distributed through the polyprotein for either of the proteases. So I mentioned previously that there are two, one located in NSP3 and one in NSP5, sorry. And we introduced these sites and then did basically the same. I will not show all images here, you can find that in the paper, but what is important is that construct number 14, which harbored the cleavage site sequence between NSP15 and 16, which is cleaved by the 3CL Pro in NSP5, gave us the highest specificity indicated here by absence of a nuclear translocation in uninfected cells, while a high number of infected cells when the virus is present. We then once again did a time course experiment to check the dynamics of this reporter system and did immunofluorescence analysis targeting viral protein. And once again, we saw a specific nuclear translocation upon infection at a very early time point. So 6 to 8 hours post-infection, we could already detect positive cells. And this correlated well with the immunofluorescence staining. So now I will go more into how the cell line can be used for investigating different parts of the viral life cycle. So our lab published a nice story last year as well for SARS-CoV-2, where we looked at the changes that occur upon infection of lung cells. And one of these alterations that we observed was that the cytoskeleton is remodeled upon infection. Specifically, vimentin, shown here in red, forms a cage around the sites of viral replication, shown in green. So this is basically a cage surrounding this area. And we were interested in the dynamics of this process. So we combined our reporter cell line with a fluorescently tagged vimentin. And what you can see here is that reporter cell line is shown here and vimentin on this side. And what you might appreciate is that this that the translocation is in the same cells as where we see the cage formation. And furthermore, I will start this video again. You might appreciate that the translocation of the reporter cell, the reporter construct, occurs at the same time as the vimentin cage formation. So if you look here, we now get an increase in green signal in the nucleus and also a vimentin cage. So this reporter cell line can therefore be used to study quite early events in the replication cycle and in a dynamic way by live cell imaging. Another thing we are working a lot with in our lab is correlative light electron microscopy. So basically, this method allows to target specific areas in a cell that you might be interested in by light microscopy, so fluorescence microscopy, and then specifically target these regions in electron microscopy. So in a method that we call low precision Clam, we do not target subcellular regions, but rather we say, okay, this cell is interesting. We want to look at this in EM, and our reporter cell line can be used to basically specifically target only the cells that are infected or transfected. And I show an example now here. So this is how it looks on the Clam dishes in fluorescence microscopy. And what you might see is that, for example, these two cells here are positive or for viral presence or protease presence, also here. And then when we zoom in case of either SARS-CoV-2 or dengue virus, we, for example, can see these beautiful ultrastructure of the endomembrane system that is heavily changed upon infection. And this can really be used to study on different levels the changes that occur upon virus infection. Finally, we can also use our construct, and we showed this in the paper in a proof of principle experiment using the nucleoside analog remdesivir. We can use the reporter cell line as a screening tool. So I show it up here. We start off with ER GFP signal, but then upon infection or transfection, for example, of the protease, we would expect to gain nuclear signal in the untreated cells. But then if we, for example, want to assess an antiviral compound, or we want to study a host vector and look if it's required for virus infection, or also if we want to study the neutralizing ability of antibodies, we can check whether we get the nuclear translocation. So that would mean that the antiviral compound does not reduce virus replication, or whether we retain the ER signal, thereby indicating that the compound, for example, helps in reducing virus replication. And to that end, we combined our cell line with a semi-automated image analysis pipeline, and then did a dilution series for remdesivir. And we could then calculate an IC50 of this compound. And when we compared it to other commonly employed assays, such as immunofluorescence staining or plug assay, where we look at the infectious virus titers, we could see that this is correlating quite well. Of course, there's a difference because it's a different assay, but overall the difference is not too big. So this can really be used as a screening tool. And what currently we are using the cell line a lot for, I mentioned this previously, is to investigate host factors that might be required for the virus replication. So to that end, we basically transfect our reporter cell line with siRNA targeting our protein of interest, host factor of interest, then infect the cells and quantify how many cells are infected, therefore showing nuclear GFP signal. So, and this is the first step, basically investigating entry and replication of the virus. And then we take the supernatant and go for another round of infection in the reporter cell line, and then we can look at more later stages of the viral life cycle, such as assembly and release. Since if the release is blocked by the siRNA knockdown, we would not expect to have high numbers of virus in our supernatant here. So to summarize, I showed you today that we generated a system that is highly versatile and can be used in different assays and is based on viral protease activity. We utilize a cellular ER anchor and modular design, therefore allowing the adaptation to other protease encoding viruses, which is important, for example, right now where we have a lot of different variants present, and it would be a lot of work to adapt the reverse genetic systems to all the different virus isolates. And this shouldn't be a problem for our reporter cell line as long as the protease cleavage site is not altered. And also other viruses can be adapted, or the cell line can be adapted to other viruses encoding proteases. And finally, I showed you different applications such as live cell imaging, Clam, and screening assays for which the cell line can be used. With that, I would like to end the talk. I would like to acknowledge a lot of people that have helped me and have worked on this together with me. First and foremost, my PI, Ralf Bartenschlager, and Mirko Cortese, who was my supervisor and mentor throughout this work, and all the other people from the molecular virology and also from the imaging platform and the EMBL who help with image acquisition and analysis. And finally, I would take the last seconds to just quickly advertise that two of the authors of this paper, who are senior postdoc, have now opened their own labs and will start to generate exciting science. So keep an eye on the Bartenschlager lab, but also on these two up-and-coming stars. Thank you.
Thank you so much for this excellent presentation, Felix. So now we can have the questions. So please share your questions or your comments. Also, I have questions. Maybe I can start with mine.
Sure.
So do this reporter system affect any cellular or viral pathways? I'm wondering.
So actually, that was one of the aims of the system to generate a system that does not affect, for example, cell viability too much. So previously, other groups have shown that you can also, instead of using a cellular ER anchor, you can use viral proteins as the ER anchor. But this, for example, was used the NS4B of flaviviruses, but this really affected the cell. In our system, we do see some effects, of course, whenever you express a protein that is not normally in the cells, they will not like it, but they can be cultured quite well. And so far, we do not experience big problems for the reporter cell line expression.
Okay. Yeah, thank you. And we have also one question in the chat box. Can you make a thread-like reporter so you can do the screening in higher throughput, screening for changes in color with KOs or in knockouts or inhibitors rather than having to look at subcellular localization?
That's a very good and valid question. We were actually also discussing that in the lab. We are trying. So basically, I agree, and we agree in the group that it would be easier to have either a black and white sort of signal difference, so have no fluorescent signal in uninfected cells, and then turn on the fluorescence when it's infected. So far, we have not been able to generate such a construct. I think thread might be possible, or a quenching sort of construct. We have not generated it so far. So we have tried different things, but so far it hasn't worked out. But we are on it. It's a very valid question. And what I can say is that, of course, the subcellular localization analysis is more work, but it works quite well for our purposes. But of course, we need the setup for the system and the pipelines.
Thank you for your question, Ryan, and for the answer. And I have one more question. I know this reporter system based on viral protease activity, but I know that some viruses, such as EBNA1, resist degradation by proteasome. So I was wondering whether this reporter system could be improved to detect them too. I mean, maybe not. I don't know
So,
if I.
Can you repeat the question?
Yeah, of course. So I know this is based on viral protease activity, but I was wondering that some viruses such as EBNA1 resist degradation by proteasome. So I was wondering whether this reporter system could be improved to detect them, to maybe adding some other techniques or change the approach. I don't know.
So this is yes. I don't know if it's possible for adenoviruses, but I honestly am not an expert in adenoviruses. So what I know is that for other viruses that express a specific protease, it's definitely easier. But I cannot tell you if we could adapt it to a proteasome-dependent mechanism. So possibly can be adapted also to cellular proteases, but yeah, we haven't tried.
Yeah, but thank you. So so now thank you so much for your answers. I think we don't have any other questions now. So